amino acids, proteins, and DNA. The definition of amino acids: a basic amino group and an acidic carboxyl group, both combined together, really. It consists of a basic amino group and an acidic carboxyl group. And because it's got both of those, it's amphoteric. It's both acidic and basic properties. So here's basically the formula for it. So you get NH2, your amino group, your your R is the side chain. That's the thing you're changing. The H, the COH, the amino group, and the carboxyl group, and this hydrogen all remain the same. This is the only thing that's changing, this side chain. The definition of Zutta ion: a dipolar ion, both the positive and negative charge in different parts of the molecule. Zutta ions only exist in an amino acid isoelectric point. What is an isoelectric point? It's the pH where the average overall charge of an amino acid is 0, and it depends on your R group. So the isoelectric point, pH where the average overall charge of an amino acid is 0, depends on R group, and Zwitter ions only exist near an amino acid's isoelectric point. So for example here, you got this, so Cr, your NH3 plus, COH, low pH, while the conditions are more acidic than the isoelectric point, so the CO minus gains a proton, a H plus ion. As Zwitter ion at isoelectric point, both the carboxyl group and the amino group as ionized, forming the Zwitter ion. And at high pH, the conditions are more alkaline than the isoelectric point, so the NH3 plus ion loses a hydrogen. Definition of proteins: polyamides, basically monomers joined by amide groups, peptide links formed in proteins. Proteins are condensation polymers of amino acids. State how proteins are made: lots of amino acids joined together by peptide links. And here you go: amino acid CrNH2HCOH plus another amino acid NH2, amino group, the carboxyl group, the hydrogens will remain the same, and this R group is different to this one, R dash. Reversible reaction: you want to go from here to here, condensation, and from here to here, hydrolysis. And all that happens is OH and this hydrogen bonds, water's lost, and then your peptide link is formed, C double bond O NH, that's your peptide link, and this is called a dipeptide, two peptides. But you need to know the conditions to hydrolyze a protein. So to go from your dipeptide to your two amino acids, you need to know the conditions. Add hot aqueous 6 meters of hydrochloric acid, heat the mixture under reflux for 24 hours. Add hot aqueous 6 meters hydrochloric acid, heat the mixture under reflux for 24 hours. And that's to hydrolyze protein. Add hot aqueous 6 meters hydrochloric acid, heat the mixture under reflux for 24 hours. Proteins. Proteins have different levels of structures. The primary structure is a sequence of amino acids in the long chain that makes up the protein, the polypeptide chain. Secondary structure: peptide links form hydrogen bonds with each other. Therefore, it's not going to be a straight line chain. The most common secondary structures are spiral, called the alpha helix, or B-pleated sheet, and it's folded like a concertina. Protein folded like a concertina. So alpha helix, B-pleated sheet. Tertiary structure: chain of amino acids is coiled and folded in a way that folded in a characteristic way identifying the protein. Extra bonds form between different parts of polypeptide chain. Therefore, protein has a three-dimensional shape, which gives it its tertiary structure. It's two main types of bond holding the protein: the shape, the hydrogen bonding, stabilizing both the secondary and tertiary structure of the protein. Cytosine contains a thiol group, the SH, and the thiol groups on the cytosine lose H atoms and join together to form a disulfide bond. Join together, so the thiol groups on cytosine lose H atoms and join together to form a sulfur-sulfur bond, stabilizing the tertiary structure. Definition of disulfide bond: thiol groups on cytosine lose H atoms and join together to form a sulfur-sulfur bond, SS bond. The active site is three-dimensional, and this gives a part of the tertiary structure of the enzyme. Say two key facts about enzymes: they catalyze every metabolic reaction in the bodies of living organisms. They catalyze every metabolic reaction in bodies of living organisms, and also proteins. Enzymes are proteins, however, some also have non-protein components. Enzymes are made of amino acids, so they contain a chiral centers. Active sites are stereospecific. They only work on one enantiomer of a substrate. Definition Active sites that are stereospecific only work on one enantiomer of a substrate. Definition of inhibitors: molecules that have a similar shape to the substrate and they block the active site, therefore no substrate fits into it. Like for example, let's say you've got a bacteria and cell wall, and you use an inhibitor, a molecule that has a similar shape to the substrate blocking the active site. Block the active site to enzyme to the cell wall, so then the cell wall bursts, so therefore the bacteria bursts. What does amount of inhibition, as a random example, I don't even know if it's legit, but you hopefully get the point, why it's going to be useful to, burst bacteria, etc. What does amount of inhibition depend on? Well, how much inhibition happens depends on the relative concentrations of the inhibitor / substrate. And amount of inhibition is affected by how strongly the inhibitor bonds to the active site. Proteins. State 2 ways scientists are finding new drug molecules. Trial and error. Scientists use lots of compounds to see if they work as inhibitors for an enzyme, and they adopt any that work and then improve those, like make it faster. So they use lots of compounds to see if they work as inhibitors for an enzyme, adopt any that work. If they work, improve them, make them faster. Computers to model the shape of an enzyme's active site and therefore how well drugs interact with it. And they quickly examine hundreds of molecules before they synthesize for the right shape before then synthesizing in the laboratory. DNA is a polymer of nucleotides made of monomers called nucleotides. Yeah, it's a polymer of nucleotides. So a key fact about nucleotides: nucleotides join together to form a polynucleotide chain. Covalent bonds form between a phosphate group on one nucleotide and sugar of another, called the sugar-phosphate backbone of the chain. Sugar-phosphate backbone of DNA is formed by condensation polymerization, forming a covalent phosphodiester bond form. DNA made of 2 polynucleotide strands. Double helix, 2 strands spiral together, held together by hydrogen bonds between the bases. Adenine always bonds with thymine. Guanine always bonds to cytosine. A to T, G to C. Definition of complementary: 2 strands of DNA are complementary, therefore they match up whenever adenine base on one strand, adenine base on one strand, thymine base on the other. Guanine, whenever adenine base, one strand thymine base and then the other guanine base on a strand, cytosine base on the other. Definition of cisplatin: a complex of platinum 2 with 2 chloride ion ligands and 2 ammonia ligands in a square planar shape. Cisplatin binds to DNA, causing kinks in the DNA helix, therefore stopping the protein from replicating DNA, copying it properly. So PT, Cl with lone pair of electrons here, Cl with the lone pair of electrons here, H3N lone pair of electrons there, NH3 with lone pair of electrons there. Presence of cisplatin complex bound to DNA strands causing strands to kink, therefore DNA strands can't unwind and be copied properly, so the cell can't replicate. Basically what I just said there. So how does it work? A nitrogen atom and a guanine base form a coordinate bond with the cisplatin's platinum ion, replacing one of the chloride ion ligands. A second nitrogen atom nearby the guanine can bond to platinum, replacing the second chloride ion. So cisplatin complex of platinum 2 with 2 chloride ion ligands and 2 ammonia ligands in square planar shape. Cisplatin is used as a chemotherapy drug. Cisplatin binds to DNA in normal cells, however, as well as cancer cells. Therefore, because it's binding to DNA in normal cells, it can therefore stop healthy cells from replicating, causing hair loss, kidney damage, suppressing the immune system. Therefore, suggested to give low dosages of cisplatin to reduce the side effects. Or we can target cisplatin to the tumor. So use a method where we're delivering the drug only to the cancer cells, so cisplatin does not have a chance to attack the healthy cells. Use a method for just delivering the drug only to cancer cells, so cisplatin does not have a chance to attack healthy cells.